We do need to get a bit of an understanding of the gastrointestinal system, and this lecture is an overview of a much larger lecture in which we go through each part of the GI tract and really delve in detail into what each one of them are doing. We're not going to do that tonight, but we are going to mention a few things which are germane to what we're discussing. So when you look at the gastrointestinal process, we all know that it really begins in the mouth. And we forget that in the mouth and in the saliva, there's a tremendous amount going on. And you begin to get a breakdown of the bolus of food that is moving down the esophageal tract in the stomach. Most of what you're looking at is enzymatic breakdown, mostly of proteins, using hydrochloric acid and pepsin, but there are many others involved. And so you get what's called a denaturing and hydrolyzing of the proteins. In the small intestine, you're looking at a different situation in which you're looking mostly at carbohydrates. Again, this is never 100%. Everything is being affected, but mostly carbohydrates and some of the fats. So you're looking at a whole series of enzymes, of which trypsin and chymotrypsin are sort of the poster children for that whole group of enzymes. And so you're breaking down the oligosaccharides and the very large saccharides into the smaller saccharides and of course breaking down proteins to amino acids, and eventually the amino acids are actively transported through the cells into the bloodstream. And remember that with stomach function, you need to remember that it has two major functions. One is mechanical, so that you're getting a temporary storage of food ingredients for a number of hours, and it allows time for the digestive enzymes to actually work. Again, pepsin is a classic example. You're also getting a mechanical breakdown of food, and it creates chyme, which is almost like what it sounds like. It's just a slurry of the foodstuff that is developed, where you have a mixture of that bolus of food with the various gastric elements, gastric juice, and it's a dynamic mixture. At the same time, you have digestion that's beginning, as I said a moment ago, so that you begin to get pepsin activity. The proteins are broken down from very large polypeptides to increasingly smaller peptide entities. And of course, there are other things taking place. You're preparing iron for absorption. This is a key feature taking place in the stomach, and a whole number of various types of intrinsic factors that are produced and then secreted. So there's a regulation of the gastric contents as it moves into the small intestine that is very much rate dependent, and it's dependent on the amount of digestion and absorption. So when you're looking at a healthy gut environment, which you see on the left, you have a microbiome, the microbiota, which are in a physiological balance, mostly commensal bacteria, always with a certain amount of pathobionts. These are the pathogenic bacterial colonies that also exist. They exist in the various portions of the mucus layers, of which there are two, and within the lumen of the gut itself. Normally you have a healthy epithelial barrier where the tight junctions are not in a very open grouping, and in the lamina propria you have the various portions of the innate and the adaptive immune system ready to be able to deal with any wayward foodstuffs or bacteria that get into the wrong place, so that they can be complexed and allowing for phagocytosis and destruction of those. If you have an unhealthy gut environment, which you see on the right, and that can be due to any number of things: antibiotic overuse, the diet itself, various types of hygienic issues, pollutants, viruses, it's endless. What you end up with in most of these cases is a form of dysbiotic behavior, a unbalanced microbiome. There's a decrease in the commensal bacteria, an increase in the pathobionts, and amongst many things that happen, you damage the epithelial barrier. And that increases the ability of bacteria to enter into the other side, the lamina propria, where they should not be, and that sets off a cascade of immune function. So having given you that very quick overview, let's really get down to some of the things that go on and why we are utilizing some of that physiology in order to build a particular test that's dealing with metabolic function. When you have normal protein metabolism, as I mentioned a few moments ago, you have proteolysis, and proteolysis is classically understood to be the taking of the proteins with various enzymatic action turns into polypeptides, and with various peptidases will eventually turn into amino acids. That's what should be happening. However, that is not what happens in many cases, and you can get abnormal protein metabolism where amino acids like tryptophan are produced in higher amounts, especially in bacterial cells, which use the amino acid tryptophan as a precursor to everything they need in order to survive. So they use tryptophanase, which is an enzyme in the bacterial cell, and you end up, excuse me, with a relatively toxic product called indole, as well as a number of other byproducts, cadaverine, putrescine, etc. We're going to concentrate on indole. I'm just going to say, excuse me for a moment. So we're going to be looking at indole because it is an important thing to see. And one begins to get an understanding of that when you look at various types of tryptophan disorders. I remind you again, tryptophan is a classic amino acid, is an essential amino acid, and is utilized by bacteria as well as human cells for their particular metabolic needs. So if you end up with normal tryptophan, it's always going to produce a little bit of indole, and that's going to go into the feces. But in abnormal situations, what is going to happen is that you're going to create excess indole, and that is reabsorbed from the intestine into the bloodstream. It gets to the liver, which creates Indican, and we're going to go through that in more detail in a moment, and eventually ends up in the urine as what used to be called indigo blue. That's the way you would actually see it. And Indican is really a biomarker of incomplete protein breakdown by mostly gram-negative bacteria and shows up in the urine. So increased urinary excretion of metabolites like Indican and 5-hydroxyindole acetic acid are what one sees in many dysfunctional situations. So why Indican? Indican is as old as the hills. We've known about that for a long time, but it is not usually tested for in dried urine, which makes it a very easy way to be able to test for Indican. And the fact that it's been around for so long doesn't make it a poor biomarker. It makes it a very good biomarker, which is very, very well known to give us a very good idea of what is happening to the digestive system in general. So remember, normal function, small quantities of indole will be formed by bacterial metabolism, metabolizing the amino acid tryptophan, and most of it is excreted into the feces. Gut dysfunction or dysbiosis, you get anaerobic intestinal bacteria that are in greater numbers because of the dysbiotic behavior. They will decompose the proteins in an incomplete way, causing what has traditionally been known as putrefaction. And it is that element of putrefaction which we are seeing in the urine when we see Indican. Much higher amounts of indole are being produced. So, Let's look at it in detail. You get partially digested food, including tryptophan, in the intestine. The anaerobic bacteria, utilizing, as I mentioned a few slides ago, tryptophanase, will break down the tryptophan into indole. Indole then goes through the portal circulation from the intestine into the liver, and the liver then begins to break down indole in a series of steps, which include it going from indoxal to a conjugated form with sulfur to indoxal sulfate, and then potassium is added to it, and it becomes the big name indoxal potassium sulfate, otherwise known as Indican. And you can see why we call it Indican and not indoxal potassium sulfate. And that's what ends up in going through the kidney, ends up in the urine, which is where we're picking it up. And so elevated urinary Indican is due to a whole number of different issues. We've mentioned dysbiosis now. You also can get it from excess protein intake. If you're taking a lot of lectins, and many of you know about lectins, you know about peanuts, lentils, various meats, various grains like certain wheats will produce high levels of lectin. That in itself will end up producing the indole, which becomes Indican. More to the point, poor protein digestion, as I mentioned a few moments ago, such as having much lower stomach acids, or for that matter, almost no acid at all, but definitely with hypochloridria, which is low stomach acid. It's not breaking down the proteins enough to begin with, and that together with what happens then in the intestine will lead to what I mentioned earlier. Intestinal permeability, where you're beginning to get a lot more parasitic and fungal infections, a lot more toxins, that will lead to Indican. Of course, protein malabsorption. There's no question that if you're not absorbing it properly, ends up sitting in the intestine where it is a prime candidate for the type of gram-negative breakdown that we were talking about. And there's no doubt that in the intestine, you have pancreatic and bile insufficiency, which is not allowing the further breakdown of the of the tryptophan, etc., leading to indole production in higher amounts. And of course, SIBO, which is a small intestinal bacterial overgrowth. So there are many conditions that will therefore elevate urinary Indican levels. A classic one is inflammatory bowel disease, which does that, including many other things. Celiac disease, which apart from the issue that we know about celiacs and their inability to be able to eat certain types of gluten, etc., it's much bigger than that. And one of the things is their inability to break down the proteins properly, the hypochloridria, as I mentioned earlier, etc. So what if the urinary results are elevated? What does one do? Well, it's really, remember, these are non-diagnostic tests. And I should have said that in the beginning. I will at the end. They are non-diagnostic, but they're still very important because it is really a starting point, if you see there's a problem with that, for further investigation into the gut health. And that is extremely important. So some of the possible directions that one can go into is, of course, trying to optimize the level of stomach acids, because I said hypochloridria is one of the prime reasons for developing this issue. You want to replenish digestive enzymes because quite often it is the lack or low levels of digestive enzymes which lead to this problem. You can get that with both prebiotic and probiotic supplementation, and of course further testing, SIBO testing, a comprehensive digestive stool analysis. All of that will help. And what you're going to see on the reports that we have is Indican shown on a horizontal bar where you get the quantitative result in the box at the top, with the same color as where it happens to be in the whole reference range area. So if in this case it was in the low reference range, you're going to see an actual value, and it's going to be sitting in a box that has the color green. So some of the conditions that are therefore resulting from digestive imbalance or dysbiosis is constipation, of course, diarrhea, bloating or gas, fatigue, very important, and a number of skin growths, which you can see with great relationship to the gut dysbiotic behavior. I'm not going to spend a lot of time on that. I do want to get to the second part of our test, because there are three parts to this test. So the first one is Indican, and the second one we're going to discuss right now because it deals with liver function. And liver function, we could spend the whole day discussing liver, but you can really break up the various aspects of liver function, of which there are over 500 that are involved in functioning for metabolic processes. But you could put them in four major categories. One, of course, is metabolism. The liver is a key aspect of metabolism in terms of synthesis, the amino acids we talked about, the proteins, cholesterol, for example, the production of bile from cholesterol, etc. Storage, one of the prime areas for storing, as you know, proteins and fats. Glycogen is a classic, and as well as vitamins and some of the key minerals. We know that it has a part to play in immunity. It is a key area for destroying bacterial pathogens coming from the gut. It is doing that constantly. And as far as the blood is concerned, we do know that it is always deactivating toxins. It is the filtering area of the body where a lot of these toxins do come in. It creates a lot of our clotting factors, and of course it will decompose many of the old and damaged red blood cells, and in many ways is a major area for the regulation of glucose, let alone fats and amino acids. And liver dysfunction can be caused for any number of reasons, most of it just being daily life and the various overloads that come from it. So if you're taking prescription and over-the-counter drugs, if you're constantly having chronic or even acute infection, the kind of food that we eat, especially processed foods with the very high sugar content, stress, both physical and psychological, especially chronic stress. I cannot, excuse the pun, I cannot stress that enough. It is incredibly important, and so on. This is the important slide. Excuse me again. Just going to take a bit of a drink. For those of you who have studied liver at all, you know that one always talks about the liver pathways, looking at the two major phases within the liver itself, which begin, in this case, with toxins, which are inevitably fat soluble, and you get phase one, which is the classic breakdown, the beginning of the breakdown of the toxins, or for that matter, anything that's getting into the liver, in which you are utilizing the reactions the body is able to harness, oxidation, reduction, hydration, etc., the utilization of many enzymes and cofactors, the many vitamins, folic acid, of course, glutathione, and the flavonoids. In that phase one, the breakdown allows you to get to what we know as the intermediate metabolites, which are becoming more water soluble. And when they become more water soluble, it allows phase two to begin to operate, and that is neutralizing whatever is left of the toxin in order to make it, allow it to be able to go into the rest of the body. And the reactions are very well known: glucuronidization, sulfation, conjunction with glutathione, of course, and with various amino acids. When you have the excretory derivative, it is now water soluble. Remember, it began fat soluble, it is now water soluble and can do what we call phase 3, which is to allow it to go into the bile, leading it into the intestines, and then out through the feces. Another way is to go into serum, into the blood, then to the kidney, and through to the urine. And so if you want to test for liver function, again, as a non-diagnostic biomarker to tell you how well detoxification is taking place and how well the liver is functioning, one of the classic tests has been testing for bile acids. And we test in this particular profile for the 12 types of bile acids, beginning with the primary types, which are known as cholic and chenodeoxycholic acid. Those are the two primary bile acids. And bile acid levels in feces and blood and urine and bile, etc., are classic markers for various diseases, which I'm not going to go into in detail. And of course, bile acids can also exist in the sulfated form, where they are also tested. We do not in this profile test for the sulfated form, but I just want you to see there are many, many different types of bile acids, beginning with the two primary ones, which are produced in the liver. And so in the liver, two primary bile acids, these are then conjugated classically with taurine and glycine. These conjugated bile acids go through the bile into the gallbladder where they're stored, or sometimes directly into the intestine without needing to be stored. And it is in the intestine that the secondary bile acids are created for the most part, so that the initial two bile acids end up with the classic 12 bile acids. And so you have these 12, the two primaries, and the dehydroxylated ones, which form 4 conjugated ones, and those give you 8 different conjugated bile acids. So the total is the 4 and 8, which makes 12. And total bile acids are a marker for screening and prognosis, for that matter, of a whole number of things. And I'm just mentioning a few here. Various types of hepatobiliary disease. And if you want to follow up on it, that's a very quick way of being able to do it. Or if you want to look at cholestasis, problems within the liver and the bile, this is a very good way of doing it. An increase in TBA, that's total bile acids, will indicate very clearly the risk for any number of things, including things like hepatitis, especially viral hepatitis, cirrhosis of the liver, drug-induced liver injury, cholestasis, etc. So it's a very valuable test, which again has been done for a very long time. And I'm not going to bore you with how it's done. There is a detection principle which starts with bile acids, and you're able to determine through a step-by-step process the quantitative measure of total bile acid concentration in the urine. And remember, we do this with dried urine. So those are the first two. Indican for maldigestion. Second one are the 12 bile acids, which tests for liver function in general, and more specifically for liver detoxification or liver detox. Extremely important. And now we're going to get to what I would arguably say is probably the most important of the three. And in order to understand that, you need to take a look at a very colorful slide which shows you a schematic of things that go on in the human cell. Please make sure you remember there's the nucleus of the cell. You've got the various cytosol, the cytoplasmic area. You've got the energy producing portions of the cell called the mitochondria. And then you have the endoplasmic reticulum, the area where you get a lot of protein breakdown, and of course the membrane, which is made mostly of lipid or lipid conjugates. So what causes cell damage? Because that's one of the things we want to look at. Well, the list is very large. Everything from radiation, smoking, I don't have to tell anyone, that's very big. Pollution becoming more and more of an issue. And of course, inflammation, metabolism itself, just the pure fact of living is creating various types of oxidative problems, which we'll deal with in just a moment. So a major cause of cell, and for that matter, DNA damage within the cell, is what we call oxidative stress. So a lot of people talk about oxidative stress. What is it? And how can we define it? Well, you need to define it by beginning with an understanding of what a free radical is. A free radical represents any entity, any species that contains one or more unpaired electrons, and it participates in a biochemical reaction. So we know that in the mitochondria, every millisecond of our life, we are producing oxidative elements, meaning that there are entities that either have one less electron or they're trying to get reduced to receive an electron, and that is called an electron transfer or a redox reaction. And remember why all this is happening. Because in the mitochondrion, you need to be able to liberate oxygen, which is part of a cycle that allows you to develop a phosphorylated entity that is going to give you the gas. It's the gasoline that's going to be able to offer the energy that the body needs. So it is part of life that you are going to produce radicals. So free radicals are being produced all the time. It's part of normal metabolic processes. And luckily, of course, the way the body works, it says we know we're going to produce these. So we have antioxidants that are going to neutralize these free radicals that are being produced all the time. So what's the problem? Well, the problem is that if there's an imbalance in that, then you get reactive oxygen and nitrogen species, ROS or NOS. And that's the imbalance in the system, the body's ability to detoxify all of these reactive intermediates that are out there. So it needs to detoxify and it needs to repair the damage that is being caused. And if it doesn't do that, you get classically what we call oxidative stress. And oxidative stress then is an increase in these ROSs, these reactive oxygen species, which again, I repeat, is happening all the time. And it shows itself in many different forms. Hydroxyl radicals, hydrogen peroxide, superoxide, you name it. There are many. So those are the sources. And they come both from within the body, endogenous, and outside, which is called exogenous. So endogenously, as I already alluded to, is just the natural fact of living and breathing. It's mitochondrial respiration. The commensal bacteria are producing it. There's metabolism of substrates within the cell as it's oxidizing, etc. And then there are people that are producing even more because they have chronic inflammatory disease, diabetes, you name it. It's also being produced outside the body and gets into us as various types of toxins. And the effects are both in the ability of the cell to be able to signal properly and not start to break down so there's a lack of communication. And there's an actual oxidative damage that can start to take place in the cell itself, in the various macromolecules making up the cell, in the lipid membranes. And I'm going to say them now because we're going to go through these. And the proteins and DNA. Those are the three major ones. There's also the RNA and various carbs that are lying in various forms within the cell. And free radicals you'll see everywhere. You're going to see on your PDF, you can go through this in detail. It's literally almost anything you can think of. But we can create a diagram of this that says, as I said a moment ago, if you're looking at oxidative stress and the cellular damage that causes, and the markers that you might need to pick up this cellular damage, what would the sources be? Well, I just said, endogenous or exogenous types of toxins, or an antioxidant dysfunction. There's just not enough antioxidant to balance out the oxidative issues that are taking place. With that, oxidative stress. And the effects are taking place in the DNA through oxidation, in the lipids through peroxidation, that's the membrane, the proteins through oxidation, and even glycoxidation. And the markers that you're going to have for that are a myriad, and I'm letting the cat out of the bag to tell you we're going to be talking specifically about 8-OHDG. And so let's go a step further. You have the reactive oxygen and nitrogen species. Through chain reactions, they cause oxidative stress. They now can damage the membrane, mostly lipid. If there's an antioxidant depletion, you're going to end up with higher amounts than you should have of lipid peroxidation, oxidative damage, mostly to the protein and the DNA breakage due to oxidative damage. And that can cause pyroptosis during chronic infection, necrosis, which is an endpoint of that kind of infective issue, or apoptosis, which is a cell's inability to continue living, and it says, I'm dying and I'm dead. Get rid of me. So if you're looking at DNA oxidation, I'm not going to go into great detail, but bacteria, both commensal and pathogenic, can affect the nucleus of the cell and actually start breaking down parts of DNA. It can do it directly, or these bacteria can do it by affecting the mitochondrion, which in turn can then begin to affect the DNA strands, mostly, as I said, through oxidation. And we're going to go through that in a few minutes. Well, I mentioned there's a second one. There's lipid peroxidation. Well, reactive oxygen species are going to oxidize the polyunsaturated fatty acids. Those are the fat entities that are sitting in the membrane and are really representing a very large percentage of what makes up the membrane. And so lipid peroxidation creates a chain reaction as this is breaking down. One of the things it does is it creates more free radicals. And when that happens and you start breaking down the membrane, you now have a lot of trouble in maintaining what's called the concentration gradient between the outside and the inside of the cell. That is a death knell. If you cannot maintain the concentration gradient, it's downhill from there. Excuse another pun on the gradient. And so what you get in that case is an increased membrane permeability, which causes inflammation, and the cell tries mightily to overcome this trauma. Sometimes it can, most of the time it cannot. The third of the three is protein oxidation. We're not going to spend a lot of time on this, except to tell you that there are two main ways in which this works. Most of them are irreversible. Oxidative modifications of the proteins. Mostly what happens is you get carbonylation of proteins. I will not go into the details of what that is, but I can tell you it ain't good. And utilizing nitrotyrosine to create complexes which are very difficult for the cell to deal with. So what do we have then? What are the triggers of a decline in mitochondrial function? Because you need to understand that oxidative damage and cell damage ultimately refers to what's happening to the mitochondria. Whatever else is doing it in the cell, it ends up affecting the ability of the cell to create enough energy to keep itself alive. And if it can't do that, it signals that it needs to destroy itself because it cannot continue. So the triggers for this decline are myriad. Stress, of course, poor nutrition, negative lifestyle factors, toxins, aging. We could spend days just discussing all of this, and I know many of you are in this field. By having all these triggers, some or most of them, you end up with decreased efficiency of the mitochondria. There's increased oxidative stress. There are more free radicals being produced than should. And there's less of the normal oxidative phosphorylation, which, as I mentioned earlier, is the way in which the mitochondrion wants to function normally. And there's damage. There's damage to the mitochondrial DNA. And I'm just going to remind you, the mitochondrion is actually a bacterium, which billions of years ago entered the cell when the cell first began to develop. And this is literally the fundamental mating or bringing together of bacteria and human cells. There are bacteria actually living in our cells. We call them the mitochondria, and they in fact allow us to live. And I want someone to ask me this question at the end. So you have increase in mitochondrial DNA corruption, increased lipid oxidation, like I mentioned earlier, and increased protein damage. All of that leads to dysfunction in its myriad of forms. You're losing that membrane potential in the mitochondrion, decreased ATP. That's the gasoline that we're supposed to be producing. You get less of it. And with all of that, you are uncoupling the mitochondrial function from the rest of the cell. And so we do have very many relative biomarkers for any number of things, including age-related diseases. And I'm just giving you a small number of these, and you can see that there's an 8-oxo-DG that's showing up in a number of these, and that's ultimately what we have decided to utilize as a marker. But you can see that oxidative stress shows itself in any number of diseases that we know very well, including, of course, COPD, diabetes, cardiovascular disease. The list is endless. We already discussed this. I won't do it at this time. And in fact, as I mentioned just a moment with CVD, oxidative stress is an important mediator of cardiovascular disease, one of the most important, in fact. And if you take a look at all of these things, from hypertension to obesity, and of course diabetes, which is a part of the aspect of obesity, smoking, renal failure, etc., these will all produce reactive oxygen species. These will be the biomarkers for oxidative stress, and that, together with a number of other things, including genetic susceptibility, lead to the various forms of cardiovascular disease. This is a busy slide, but an important one, because it shows the importance of oxidative stress in degenerative diseases and in cancer. And what we're looking at here is, with aging, very often due to things going on in the environment and the internal effects in the cell itself, you get mitochondrial dysfunction and a beginning of a breakdown of regular metabolic function, otherwise known as metabolic deregulation. That is causing inflammation. Inflammation causes oxidative stress, which is showing itself in the areas which I told you: DNA damage, lipid peroxidation, and protein oxidation. So that's a common pathway for most of what we see. But DNA damage can also cause mutation, and inflammation can also cause tissue injury, which then can lead to distinctive pathways which will create abnormal protein aggregation, which the cell says, I can't handle, and that leads it down the apoptotic pathway, the pathway to cell death and the generation of the cell. But you can also get tissue injury, which then will form stem cell activation, and that, together with mutation, can create mutating cells that proliferate, and that causes carcinogenic activity. I want to make sure that I don't miss telling you about the intestinal barrier and the protection against oxidative stress. You need to remember, as I said, that oxidative stress, amongst other things, causes damage to the proteins in the cells, mostly through carbonylation, and that is irreversible, so that you get a loss of protein function. And the result of that loss of protein function is a breakdown in the integrity of the tight junctions between the intestinal cells. And so that means you end up with increased intestinal permeability. I don't like the word leaky gut, but many of you have heard that term. And here we see the difference between a healthy gut with a lot of mucus, and here we see on the unhealthy side that a lot of bacteria and toxins, including lipopolysaccharides, which, if you remember, are the membranes of gram-negative bacteria, enter into the wrong side, the other side of the barrier. And with that, something you don't talk about very often is that you're also getting a very dramatic increase in reactive oxygen species, these free radicals. And that leads to oxidative stress, a very hyper immune reaction. And I don't have to tell you that that is a downward slope. So apoptosis, as I mentioned earlier, is programmed cell death. The cell says, I'm going to die, remove me, replace me with a new cell. So, number one, a cell gets damaged, it's stressed or it's triggered in some way, and it activates the signaling cascade to destroy itself. And it begins by shrinking, and then you get the series of caspases. Some of you may have that biochemical, you did courses on that, and you'll know about the caspase system, which is activated, and it breaks down the various internal cellular components, and that will lead eventually to a whole number of things going on, including enzymes that literally break down the nucleus and fragment it, and that is a signal that brings in the macrophages, which are there to eat up the broken pieces of the cell, and that is the apoptotic endpoint. That's the end game in apoptosis, which is macrophages getting rid of the components. And so there are a whole number of biomarkers that are used for oxidative stress, and the one that we have decided to use for a whole number of reasons is 8-hydroxydeoxyguanosine, known lovingly as 8-OHDG. It's been known for quite a few years, and it works as a very stable biomarker in cells and tissues, in blood, in urine, etc. And so we are looking at DNA oxidation, which is really the classic way of wanting to look at what happens when a cell is damaged and is going through oxidative stress. And we know that 8 OHDG, which is what we're going to call it, is a biomarker of cumulative cellular oxidative stress. It's not an acute marker, because an acute marker can change minute by minute. That's not what you're looking for. What you want to have is a non-diagnostic test that tells you how well is that person functioning and how damaged are their cells. So it's really a way of quantitatively assessing ongoing oxidative damage. And of course, in that way, you're able to monitor the effects of various types of antioxidant therapy, whatever you're doing, whether it's with supplements, whether it's with diet, or both, and what you're doing to try to detoxify the liver, etc. And as I said, we use a simple collection system, which is dried urine. And so we can say then that cellular 8-OHDG concentration, for a whole number of reasons, is the established marker of oxidative stress. And we know that oxidative stress and reactive oxygen species induced urinary elevation of 8-OHDG is associated with a whole number of pathological processes. I'm not going to bore you with this. It's pretty obvious. You're looking at here at the names that we all know very well, from inflammatory bowel disease and various types of metabolic syndromes, etc. And this is another very busy slide that is quite important, because it really says to you, what does persistent oxidative damage, the lesions caused by oxidation, how does that create the endpoint of apoptosis or cancer? And so what we're looking at here is a cell that is being affected by both endogenous sources, meaning various types of oxygen metabolism, inflammation, in other words, the stuff of life, that together with exogenous sources. What are they? Well, radiation, UV radiation, chemicals, drugs, smoking, folks, very important. All of that makes the cell produce an excess of reactive oxygen species. Now, luckily, normally, as I said, there are radical scavengers. They take care, they neutralize these radicals. Glutathione, of course, is the poster child for that, and many supplements for that you utilize many different types of antioxidant enzymes, including superoxide dismutase, etc., catalase, SOD, CAT, etc., many vitamins, and that will manage to keep the DNA functioning with very little damage. But damage does occur. And oxidative damage, when it does occur, can occur either as an isolated damage to one of the base pairs, or it can be a series of clustered DNA lesions which can take place. And in most of these cases, you are oxidizing the bases to be able to create what's called 8-OHDG. You are oxidizing guanosine, which is one of the 4 bases that makes up the genetic code. And that, of course, can be repaired to a certain degree. And, of course, in some cases, you will repair DNA damage. If you don't, then if there's very persistent damage, you'll get either apoptosis, as I said, that is the death of the cell, or you're going to get some kind of mutation and a replication of that mutated portion of the DNA, which is going to create ultimately a malignant or a cancer cell. Just a reminder, there are many different types of antioxidants. The endogenous ones, very well known. Here are the very important ones, the catalases, superoxide dismutase, the glutathione peroxidase, and glutathione reductase. These are the classic endogenous antioxidants. And of course, you all know very well that you can get a lot of exogenous antioxidants, not just in the diet, but of course through supplementation with the various types of flavonoids, quercetin, resveratrol, curcumin, etc. And so when you're looking at the interaction of nutrition, of antioxidants, of oxidative stress, and general health status, you can look here in this slide on all of the different reactive oxygen species that can cause problems. What are some of the diseases that it will cause? What are the antioxidants that we can utilize? And ultimately, what are the biomarkers that we can use? And we have chosen, for the reasons which I mentioned earlier, have chosen what is known in the literature as one of the most stable biomarkers for cellular oxidative stress. And so that was my very quick overview